we've got a staining rack here.
Yeah.
And this is 2 pieces of rod joined simply by a small piece of rubber tubing.
Okay.
And they're very easy to make in the classroom. You just need to make sure that the piece of rubber tubing here fits nicely so that when you take a microscope slide, then that will fit onto the rack that you've made there. Now the secret of good slide making here is to have new clean slides. Why do you think we're going to have good clean slides?
I'm assuming that if there's a lot of dirt on the slide, then you maybe get some residual staining or something like that?
Absolutely.
Yeah.
If we've got a dirty slide, then we get poor staining.
Yeah.
Also, of course, it's difficult to spread the loop of water over.
Oh, of course.
And it just kind of comes together.
Can I just ask you, John, you said that you just need to make this with a couple of rods. You've got a metal rods here. Would glass work?
You could use glass.
Yeah. Issues with breaking, I guess, and that.
Sometimes if you've got pupils that are a bit ham-fisted, they'll break it, and therefore I've just got some nice metal rod.
poles.
But again, you could use glass ones.
Great.
I've got a culture of microorganisms here, and we can't tell very much about the structure of them, and we can't actually identify them. But I happen to know, and I'm pretty confident, that it's a pure strain that we've been growing in the laboratory here. But what we can do is we can make a slide, and we can stain the slide, and that will tell us something about the organisms. But if we wanted to actually identify it, what we would have to do are some biochemical tests that are not complicated, but often cost a bit of money. And so what we can do is we can simply stain them in the laboratory and find out more about them.
Okay. One of the stains that we could use is just a simple stain, which would be very simply just fixing the microorganisms onto the slide like we would do for any technique like this. And we would just stain with a red dye called safranin. And safranin, we would use that principally just so that we can look at the cells underneath the microscope and we can tell what shape they are.
Yes.
So as we know, when we look down a microscope, you can see that we have lovely cocci, so lovely round cells, or we have rods, which are shown as little rod shapes. And then there's various other morphologies, so shapes that we can look at. Now, the safranin doesn't give us much more information than that. There's another staining technique that would give us a lot more information.
Now, I think you're talking about gram staining.
Yeah, that's right. Yeah,
and what we can do is we can take the organisms, and this time we can stain with 2 other stains.
Yeah.
And if the cell wall around the organism is quite big, then what tends to happen is that this stain gets caught in the structure.
And what colour is this
Now,
stain?
this is crystal violet.
Right.
And we use the iodine. And what happens is we form a crystal violet iodine complex. Technically, the iodine here is a mordant, so it fixes the crystal violet into the wall of the bacterium. Now, if we've got one group of organisms, then when we wash with alcohol, it will wash out. But with the other group, it remains in the wall. So if we've got a thick wall, it remains in there. If we've got a thin wall, it washes out.
Okay.
But we've got a problem. For half the cells that we stain, if you've got a nice thick wall, they stain nice and purple. But for the others, they become colourless. So we come back to your simple stain.
Ah, okay.
We now use this stain again. If they're already purple, we just don't see it. If they've lost the stain, then they'll be nice and red and we can have a look at them. So the first thing we need to do is to get some of our organisms onto a microscope slide.
Okay.
I'm going to turn the Bunsen so that we've got a little raw, and I'm just going to sterilise the loop before I start. Flame it, and I'm going to take a nice loop of water.
Is your water sterile?
This is actually sterile, but it doesn't need to be.
Okay.
We could just use ordinary tap water because the number of cells in the tap water are so few. I've got a nice blob of water there. Now what I'm going to do is to sterilise the loop again and to take a few cells from this agar plate. To test that it's not too hot, I'm just going to touch the agar, and then I'm just going to pick a colony So I've just got a very few cells on the end of that inoculating loop.
Now each colony, how many microbes do we think are in a colony?
There's probably going to be millions of
Millions
cells.
of
Millions
cells.
of cells.
Okay.
And now I'm just going to streak the loop all the way through that blob of water and spread out the small pinhead of cells over an area about, well, it's going to be about 1 centimetre by 2 centimetres, spreading it as thinly as I possibly can. Now this takes a little bit of time. Here we are. When I finished, I'm just going to sterilise the loop, place it down there. I don't need that, so I'll just turn that back to a yellow flame and move it slightly out of the way. Now we need to leave that for a moment just to dry. So while that's drying, I was thinking that I might be able to do another slide because I believe that that's a gram-positive organism and this one is a gram-negative organism.
Okay.
Therefore we could have a look at the end. So again, I'm just going to sterilise this loop, hold the top, flame the neck. I'll take a loop full of water.
If you were going to do this with a broth, so we've used broth cultures up until now, I mean, does that make a difference, or what would you do differently there?
I wouldn't do anything different. Sometimes the problem with a broth is that you get a lot of the broth onto the slide, but you can use a broth. It wouldn't make a lot of difference. Let's go back to this loop here. Let's just sterilise that. Touch where nothing is growing, and then again take a small loop of cells. I'm going to spread that out.
Is it better that we have lots and lots of bacteria to make sure that we see something, or should we put on less? How much should we put on?
This is a classic case of where fewer is better.
Okay.
If you have too many cells, when you look down the microscope, they're all mashed together and you can't see the individual cells and therefore the structure and whether there's a kind of format to those cells.
Okay.
Now that we've got the cells on there and they have dried, what we need to do is to fix them. And we're going to fix them by passing them very briefly through that flame. And it's really just an action of straight through the flame like that. A little bit of heat and the proteins will coagulate and it will fix the cells to the slide. So I've done that slide and now I'll do this one. I just pass it through the flame fairly quickly so there's just a little bit of heat.
And you're passing it through with the cells on the top of the slide and the heat coming from the bottom,
Absolutely.
Is that right? Okay.
If I did it the other way, we would damage our cells.
They would be a bit frazzled. Okay.
Yes. Now we're ready to stain. Now we're going to, let me just turn that Bunsen off and then I won't burn myself. Now what we're going to do is to stain these cells with the crystal violet. I just put a few drops on, making sure that I cover the smear from this end right the way down to that end there. And I need to leave that on there for exactly 1 minute. During that 1 minute, the stain is going to get into those cells. Then, at the end of 1 minute, I'm gently going to pick the slide up, allow the liquid to drain off, and then add the grams iodine.
Okay.
Now, I've got a very cheap bowl here, because one of the problems is that if you use a sink, you soon find that you get purple dye, yellowy brown dye and red dye all over the sink, and someone spends ages trying to clean it.
Yeah.
If we use this, then everything is contained within that, and we can wash it away at the end. We can dilute it down and wash it away so the sinks don't get stained. I think that has been almost a minute. And what we're going to do now is to wash off this stain. I tip the slide and I gently squirt some of this iodine solution over the top. Just checking that at the bottom there it's gone. And then I'll just put a few drops on the top like that. I'll do exactly the same for this one. Squirt the iodine solution over the top, check that there's nothing on the back, and then leave the stain on. And now you can see that the smear that we've made is changing colour. It's gone a lot darker in colour. We need to leave that for a minute. Now that's the end of the second minute. 1 minute for the crystal violet, 1 minute for the grams iodine.
Okay.
Now we're going to wash off with alcohol. So again, just as I did before, I'm gently going to squirt the alcohol over. And what you'll notice is we've got pale blue washings coming off. Now we need to do this so we get rid of all of the stain, but if we get too carried away, we'll wash out all of the stain. So I'm now just gently going to wash that one off.
Is that 100% alcohol?
Yes, this is 100% alcohol. That's looking okay, and I'm just going to take the last bit of that off there. Okay. Now what we need to do is to stain with this rather nice red stain that you said earlier on we could use for a simple stain.
Sure.
And I'm now going to gently pour that on and leave that to sit there quite happily. Good. And normally I would leave that there for about 30 seconds. At the end of 30 seconds, we'll wash it off with a small amount of water. Now I think that's been 30 seconds, so we can proceed. I'm gently going to tip the slide to get rid of the stain, and then I'm going to wash off with this.
Now John, you said that the cultures that we're using are pure cultures. You said that earlier. Can you use this for identifying contaminants, or
yes, you
if
could.
You're ill from body samples or anything like that?
Well, I wouldn't suggest if you've got a terrible cold that you start taking samples from yourself and doing this. But if you suspect that a culture you've got isn't what you thought it was, then yes, you could do this. You could take some culture, you could make a slide, and if suddenly you see there are lovely rods and beautiful cockeye, oh dear.
We know it's a mixed culture.
You know it's a mixed culture and you've got a contaminant.
Okay.
If you see just one type of cell, then okay, if it's supposed to be a rod because you think it's going to be E. coli, then that's a clue that it probably is. But again, it could be another rod. It could be something else. But again, it's indicative that you've probably got a culture that's okay.
Okay.
Now that we've washed that off, we need to just blot it dry. I've got some nice blotting paper here, and I'm going to put that there, put that one there, and then just put another piece on the top. Now this is important because if we don't get rid of that water, that moisture, when we try to look at them under the microscope, because we're going to use oil immersion, the oil won't give us a decent vision. We won't be able to see clearly what's there. The oil will mix with the water, we get a horrible emulsion, and it's not going to look good. Now I think that's dry, but just one more to make sure. Yeah, that looks beautiful.
Yeah.
Now we're ready to look at these under the microscope. Of course, I happen to know that this one here was Bacillus subtilis, and this one was an E. coli. And it's worth labelling so that we know when we look at them under the microscope which is which. And
it's probably better to label afterwards, am I right in thinking that?
I tend to do that because often the alcohol and the stains get all over it.
Okay.
But again, if you do label at the beginning, just check that the name is still there.
So if you're doing it in the classroom, just make sure you try and keep track or just do one at a time or something like that.
Now these are ready to have a look under the microscope.
Excellent.